Hello again. This is lesson 6, and it deals with the ecology of marine photoheterotrophic bacteria. This is actually my expertise, my field of research, and is also the last lesson that I am teaching this course. So in terms of carbon metabolism, there are 3 types of functional groups of microbes. Phototrophs, they fix CO2 from the atmosphere using light, and they produce organic matter. And the second group are the heterotrophs. They require organic matter for energy and for growth, and they produce CO2. And then the photoheterotrophs are organisms that require organic matter, but they can also use light to produce some energy. So these would be the 3 functional types of microbes based on their carbon metabolism. So until early 2000, only 2 types were considered, phototrophs and heterotrophs. But in 2000, there was the discovery of photoheterotrophs, and now we know that there are 3 kinds of bacterioplankton in terms of carbon metabolism. Well, as I said, the discovery was in 2000, and funny enough, actually the 2 types that exist, there are a system based on bacterial chlorophyll A and a system based on proteorhodopsin, were discovered with a day of difference. See here the dates. This paper was published 14 September 2000, and this one was published 15 September 2000. So in the first article, Colbert and collaborators reported the observation of distinct signals of bacterial chlorophyll A containing microorganisms in the surface waters of the tropical Pacific. And these signals were linked to the presence of what are known as aerobic and oxygenic phototrophic bacteria, or AAPs, that you'll hear a lot about them in this class. Then in the second paper, Beja et al. Announced the discovery of a new rhodopsin gene in DNA samples recovered from the Monterey Bay in California. And we've talked about this because this was discovered through metagenomics. So rhodopsins are well-known membrane proteins that serve as a photoreceptor molecules in animals, including us, humans, we have them, or they can act as proton pumps in allophilic archaea. But until Beja's report, they were not thought to be present in proteobacteria. That's why these rhodopsins were called proteorhodopsins. Well, so these are the 2 types, and there are actually only 2 types of biological systems that are capable of producing net energy or of net energy conservation. And one is based on chlorophylls, like the chlorophyll that phytoplankton has, or bacterial chlorophyll, different types of bacterial chlorophylls. And the other one is based on retinal molecules. So retinal group is the one that is excited by light, and then the rhodopsin acts as a proton pump, and this proton gradient is used to synthesize ATP. So we've seen this already. Proteorhodopsin were discovered in an uncultured bacterium from the SAR 86 group, and it was discovered through metagenomics. However, in this first study, it was thought that it was something unusual or anecdotic, and that this was not a widespread system as we now know it is. So after the first study, a second study was focused on trying to see the function of these proteorhodopsins, and basically they described the structure of the protein, and they also saw that there were 2 different families of proteorhodopsins in the sea, absorbing light with different absorption maxima. One absorbs the green light, and the other one absorbs the blue light. And it was shown that this distribution, the distribution of organisms containing these 2 types of proteorhodopsins, had to do with stratification of the the water column. And also that only a single amino acid residue function as a spectral tuning. So there's only one difference nucleotide. So leucine gives a green absorption, and glycine leaves a blue absorption for the rhodopsins. This actually, this spectral tuning seems to be an adaptation to different habitats, absorbing different light at different wavelengths according with the light that is available in each environment. So here you see the spectra of light penetrating the open ocean water column, and here in the coastal water column. So you see that there's more penetration in the open ocean than in the coastal, and also that the blue light penetrates deeper or more, penetrates more than in open ocean than in coastal waters. So this actually has to do with the distribution of these, sorry, go back, these two types. And we know now that there are more protorhodopsins absorbing in blue light in open ocean waters and in deeper waters than those that absorb the green light that are more abundant in coastal waters than in the open ocean. So after the studies by Beja, another tipping point, you already saw this also, was the sequencing of the first metagenome sequencing from the Sargasso Sea that reported almost 800 different rhodopsin genes. And here we see in blue, this blue, where the rhodopsins that were known before from eukaryotes and archaea. In yellow, other uncultured protorhodopsins. And in red, we see all these from new rhodopsins from the Sargasso Sea, and you see that it's a much more diversity than it was thought to exist before. So there are, as I said before, it was thought that it was something anecdotic, but with time and with multiple studies, it has been seen that depending on the environment, and if it's more like the open ocean or the coastal ocean or in different regions, depending on the conditions, between 13% to over 80% of bacteria in surface waters actually carry these proteorhodopsins. And this took relevance when it was when the sequence of the SAR11 genome was completed, and it was shown that actually SAR11, that is this very abundant area in the ocean, present protorhodopsin, contains the gene for protorhodopsins. And the genetic system for protorhodopsin is quite simple. So this actually has done that it has been transferred between multiple species through a process known as lateral gene transfer, which means that different taxa, like far apart taxa, can exchange genes. And that's why, probably why it's so widespread, because it's very easy to transfer, because it's only based in two genes. And this also makes that sometimes it has been transferred even from bacteria to archaea, or it's not only from archaea to eukaryotes. But that makes it very, very widespread. So the hypothesis of the working hypothesis of understanding why it's an advantage to be a photoheterotrophs versus a strict heterotrophs was initially that this provides an ecological advantage, because you have an additional source of energy, producing energy. So in environments where organic matter is limiting or limited, it would be an advantage to be able to produce ATP through light. And so it was thought that these systems would be more abundant in oligotrophic waters, and also that the advantage would be that these organisms would have higher growth rates under light than under dark, because they can produce additional energy through light. So the first experimental work on Proterodopsin containing culture marine bacteria, members of the SAR11 and the SAR92 clades, actually did not identify this increased growth that was expected for having Proterodopsins when they were growing in light. So here you see the results. You see that the growth under light and dark, it's very similar. No difference. So this, well, was a little bit disappointing because then it was not known what was actually the actual advantage of having Proterodopsin, but later studies, and in this case with the species of Vibrio, showed that what rhodopsins or Proterodopsins do is like they promote survival during starvation. So here we see a growth rate, a growth curve of bacteria. And so this is the exponential phase. And during the exponential phase, there's pretty much no difference between growing in under dark conditions or under light conditions. But after exponential growth comes to an end and they start the and they get into starvation, then you see that the survival under light, which is these open circles, is higher than under darker conditions. So one of the functions of Proterodopsins is promoting survival during conditions of starvation. Okay, so later studies with the SAR11 organisms, when it was isolated, have shown that energy-starved cells can maintain size, ATP content, and higher substrate transport rates, and they can expand differentially about 10% of their genome when they are growing under light than under dark conditions. But still, like, these are additional functions, but the main function that it was thought to occur, which is increased growth when growing under dark, it wasn't shown until this paper was published with, in this case, with species of marine flavobacteria that it's a member of the Bacteroidetes, and showed this experiment. And this experiment showed that when the culture was grown under light, the cell counts was much higher than they were growing under dark, and also that a culture that was under dark, once it was switched to grow under light conditions, it could also reach higher cell concentrations. So now it seems that actually these Proterodopsins can have different functions, and from, for example, from promoting long-term starvation, that would be strategy sorry, when cells are under carbon poor, in a carbon poor environment, perhaps the energy that it's able to obtain from light is not devoted to growth, but to maintain survival. And when they are in conditions, for example, where they can be attached to particles of the other dying cells, or they are in a richer environment, then in this case, perhaps what they do is like to increase growth rate and enhances yield. Okay, and well, after many studies, this nice review was published. If you're interested in the topic, I recommend you to read it, where it compares the bioenergetics of the different the two types of photoheterotrophic bacteria. And so it concludes that that Proterodopsins have a low gross gain of energy, but it has a low construction cost. The genetic system is very simple, so the cells don't have to invest a lot of energy in obtaining these proteins. And so this makes that they have to synthesize many Proterodopsin molecules in the cell to gain any energy from phototrophy. So the conclusion would be that the net gain is low, but it's beneficial, and so that's why it's so widespread in the prokaryotic world. Like, as I said, almost up to more than 80% of prokaryotic cells in the surface ocean can actually be photoheterotrophs of containing proterodopsins. So this part is it's shorter than the AAPs part that we will start now, but I wanted to give you an overview of this proterodopsin containing bacteria. And there are many aspects that are still not known, and there's a lot of research going on in this field. But the conclusions are that proterodopsins play different roles in microbial metabolism, that proterodopsins photoheterotrophy might stimulate growth of some marine bacteria, but also promotes the survival of other bacteria under starvation, and that the diversity is large and new rhodopsins are still being discovered. Okay, so we switch to the second type, and the second type is a bacteriochlorophyll system, which is similar than to the chlorophyll system from phytoplankton or plants, but in this case, it's a system that it's present in bacteria. In these bacteria that contain these bacteriochlorophyll A are known as aerobic anoxygenic phototrophic bacteria, or AAPs. And to summarize, they contain this pigment, bacteriochlorophyll A. They are photoheterotrophs, so they require dissolved carbon or organic matter to grow, but they can utilize light, and they respire CO2. And they actually went through these horizontal gene transfer processes, and this means that we cannot use the phylogeny of 16S, the phylogeny of the 16S to identify them, which means that, for example, two species that are very closely related in 16S, perhaps one acquired these genes, so it can be considered an AAP, and others did not acquire them. And species that are more far apart can actually have these genes in common with these other species. So we cannot just know if a species will be an AAP by sequencing the 16S, so we need to use additional genetic markers, and in this case, the PAF operon is the one that encodes for the bacteriochlorophyll system, is the one that is being used to identify these AAP species. So as this is the first paper where they were discovered, and it was big news, but it wasn't known if it was an important system, if it was relevant, if they were abundant, if they were active, and so on. So in a second paper that was published only a year later, it was shown that in the Pacific Ocean, at least 11% of the total microbial community could be this type of bacteria, AAPs. Okay, so after these initial studies, different researchers from different places started to look for them in different environments. These species can be observed in an infrared microscopy because bacteriochlorophyll A emits in the infrared, so they can be visualized if you have one of these microscopes. And so in a study in the Southern Pacific, actually carried on to test this hypothesis, this initial hypothesis known as Culver's hypothesis, saying that they would be more abundant in oligotrophic areas because in oligotrophic areas cells have less carbon, and so if they have this additional source of energy, then they would be more competitive over strict heterotrophs. And they reported that up to 24% of prokaryotes were AAPs. However, these results or this hypothesis got in conflict with other papers that were published later on that showed that, for example, they were abundant in a rich estuary, which is not it's not an oligotrophic site, it's actually quite eutrophic. So this contradicted Culver's hypothesis. And as we accumulated data, we saw that the results were quite variable, depending on the method that was used to quantify them, depending on the environment, depending on the time of the year, depending on many other factors. But in general, we can conclude that depending on the site, they can be from less than 1% to up to 25% of prokaryotes, but there were not much differences between very oligotrophic sites like the Sargasso Sea or some parts of the Mediterranean Sea than the Baltic Sea, that is a much more eutrophic environment. So to test this hypothesis, me and some colleagues, we tested this hypothesis in the Mediterranean. The Mediterranean is a nice environment to test this hypothesis because it has an increasing gradient of oligotrophy. So the Eastern Mediterranean is much more oligotrophic than the Western Mediterranean. So collecting samples from different cruises and different sampling stations, we could quantify these cells through infrared microscopy. And here you see the results. So this is the percentage of AAP cells, and this is the chlorophyll A. Chlorophyll A, remember, it's a pigment that phytoplankton has, and it's a proxy for productivity. So the more chlorophyll, the more productive is the system, and so less oligotrophic and more eutrophic. So the stations here are sorted by the oligotrophic gradient. So this would be the least oligotrophic, and Villefranche Bay would be the more excuse me. So Eastern Mediterranean is very oligotrophic, and Villefranche Bay is not very oligotrophic. And so you see that actually the percentage of AAP cells increase as the chlorophyll increases. So at least in the Mediterranean, we saw that the spatial distribution of AAPs is in conflict with Colbert's initial hypothesis. This was in agreement with a parallel study conducted by French researchers that was published the same year, and that they went through a cruise across the Mediterranean, and they also found that higher bacterial chlorophyll concentration, which is the pigment of these AAP cells, was higher towards the Western Mediterranean. So there are more AAPs in the Western Mediterranean than in the Eastern Mediterranean. So, okay, now we jump to a more global study, and this is the Global Ocean Survey, which is this study I have talked to you about in a few times. That was conducted by Craig Benter. So all this metagenomic data that they generated was put in public databases, and so people could access them and study their genes that they were interested in. So a few years later, there was a paper published in this environmental microbiology journal, and so what they did was to get all the genes that were related to the PAF operon from the metagenomic data set, and they estimated the percentage of abundance of these AAP cells. And so the Global Ocean Survey was initially conducted in the Sargasso Sea, but then it went from the east coast of the United States to the Galapagos Islands and the Pacific Ocean. And then here you can see the results, and the numbers are variable from less like 1% to up to 12%, but we can see that the most abundant, the higher numbers were recovered from coastal areas that are typically not oligotrophic. And areas like the Sargasso Sea that are more oligotrophic had less concentrations of these AAP cells. So to summarize this abundance part, to say that regardless of the method that is used to quantify them, that it can be the concentration of the pigment, it can be counting the cells through infrared microscopy, or it could also be estimating the percentage of cells through recovering their genes through metagenomics. Also, we can use a method known as quantitative PCR that we can quantify the marker genes for these organisms. Regardless of the method, on average, we can say that in oligotrophic regions, there are in generally, like on average, less than 5% of the cells, and in more eutrophic regions are usually up to 10%. But in conclusion, this shows that the initial hypothesis of Colbert was wrong, and that these cells actually are not very well adapted to very oligotrophic conditions. Okay. And then this is a very recent study, it's from last year, 2019. And it was the first study that actually compared the three systems of pigments that are known to occur in the ocean, so rhodopsins, chlorophyll from phytoplankton, and bacterial chlorophyll in a transect from the Mediterranean. Here you can see the results of chlorophyll, rhodopsins, and bacterial chlorophyll, and through depth. Okay, and so these warm colors means higher concentration. So we can nicely see that actually the chlorophyll and the bacterial chlorophyll pattern is quite similar. So they seem to be occupying the same niche. And rhodopsins actually follow the opposite pattern. So in the areas where there's less chlorophyll and less bacterial chlorophyll, there's more protorhodopsins. So that these two types actually follow the opposite strategy to occupying the different ecological niches. So, well, this I already showed that there is the Colbert's hypothesis is in conflict with the existing data, and that these cells are more abundant in less oligotrophic environments. Okay, so this was data about the spatial distribution, but we were also interested in knowing if they were seasonal, because if they depend on light, one may expect that they could be seasonal. And so we took advantage of a study site that it's near the coast of Barcelona, actually in the town of Blanes. It's known as the Blanes Bay Microbial Observatory, and it's conducted by the Marine Science Institute in Barcelona, where I worked for many years. And so this is it's a microbial observatory, so it's monthly sampled, or sometimes at a higher frequency. So we had DNA from many years, and so we took DNA from 10 years through this quantitative PCR method that I told you that allows to quantify the marker genes for these groups. We could measure their abundance through the different seasons. And you can see here that there is a seasonality, and that they are more abundant in spring and summer than in fall and winter. From these same samples, we also had metagenomics, and so only for 3 years, not for the 10 years, but we had metagenomics data, and we could extract the genes related to these AAPs, and also show that even though the absolute numbers were slightly different, because it's a different method that has different biases, the trend was the same. They are more abundant in spring and summer than in fall and winter. And nicely, a paper from a similar microbial observatory, but in this case in Bagnols-sur-Mer, which is in France. It's north of Blanes in southern France, showed also very similar trends. The order of the seasons is different here, so spring starts in here, spring, summer, autumn, and fall, but see that the trend is the same. And this study was done through metagenomics, so it's nice to see that the results match very nicely. Okay, and I'm going to try to continue with the video without stopping the video. Let's see. Okay. Two. Okay, so we talked about their abundance in different environments and spatial and seasonal or temporal patterns. And the next question I'm gonna talk about is about their diversity. So we were interested in to see how many are they and how are they distributed, and then also on who they are, what is the diversity, and later I will talk about what do they do, what is their function. So based on the 16S from isolates, we know that AAPs belong to either the alpha protobacteria or gamma protobacteria that are abundant and relevant marine bacteria. But as I said, the 16S doesn't allow us to work with them in the environment because two very similar species based on 16S can either be AAP or not AAP, and the opposite, like two far apart species through the 16S can actually both be AAP due to this lateral gene transfer or horizontal gene transfer process. So we need to use functional marker genes. And here's the same study that I talked about, the Global Ocean Survey data, but in this case showing the results about their diversity, not abundance. And the people that published this paper were actually the ones doing a classification of the different AAP types, and they did it based on the phylogeny of the PAF-M gene marker. The PAF operand has several genes, but this was shown to be the best for doing phylogenetic analysis. And so they classified these AAPs into phylogroups that they named from A to L, so A, B, C, D, etc. And then they realized that there were some of these phylotypes of phylogroups that had no culture representatives, so they were similar to nothing known. And whereas others, like phylogroup A or phylogroup G, they were similar to or members of the known species, like, for example, the Rosabacter or Rhodobacter, alpha protobacteria, or to some members of the gamma protobacteria, like phylogroup K. So they concluded that they said that the A to D groups that are uncultured were actually the most abundant from this study. That phylogroups A and B dominate in oligotrophic regions, where phylogroups E and K dominate in more eutrophic regions, and that phylogroup G, which is related to Rosabacter, is the most ubiquitous in different regions. Okay, so after this paper from the Global Ocean Survey, there were a report on the diversity in the Mediterranean Sea, and it actually contradicted the results published by in the previous paper, because it showed that that the most abundant group in the Mediterranean was Gamma Protobacteria. And this study was done with DGG and clone libraries. So at that time, it wasn't known whether this was because of the different methodology used, or because the study was done in summer, and perhaps in a different season we would find similar results to those from other places, or perhaps it was a signature that the Mediterranean actually has a different community of AAPs than other regions of the world ocean. Okay. It's not going through. Why not? Okay. Okay, it's not going through, so I will stop the video here and I will continue in the next video.